Hello dear students, welcome back to the channel all about biology. So in the previous video we studied that DNA contains genetic information required to make proteins, because in order to properly function and survive, living things must express their genes to make proteins. Today we are going to find out that how these genes are expressed. In order for our bodies to function, we need to supply them with a variety of nutrients we get from our diet. Our bodies cannot use the food as it is when it enters into our digestive system. The process of chemical digestion uses different proteins and enzymes to break down the food particles into usable nutrients our cells can absorb. The instruction to make these proteins are contained in our DNA. DNA contain genes. A gene is a continuous string of nucleotides containing a region that codes for an RNA molecule. This region begins with a promoter and ends at a terminator. Genes also contain regulatory sequences that can be found near the promoter or at a distant location. For some genes, the encoded RNA is used to synthesize a protein in the process called gene expression. For these genes, expression can be divided into two processes, transcription and translation. In eukaryotic cells, transcription occurs in the nucleus where DNA is used as a template to make messenger RNA. Then in translation, which occurs in cytoplasm of the cell, the information contained in the messenger RNA is used to make a polypeptide. During transcription, the DNA and the genes is used as a template to make messenger RNA strand with the help of the enzyme RNA polymerase. This process occurs in three stages: initiation, elongation, and termination. During initiation, the promoter region of the gene functions as a recognition site for RNA polymerase to bind. This is where majority of gene expression is controlled, by either permitting or blocking access to this site by the RNA polymerase. Binding causes the DNA double helix to unwind and open. Then during elongation, the RNA polymerase slides along the template DNA strand. As the complementary bases pair up, the RNA polymerase links the nucleotide to the 3' end of the growing RNA molecule. Once the RNA polymerase reaches the terminator portion of the gene, the messenger RNA transcript is complete and the RNA polymerase, the DNA strand, and the messenger RNA transcript dissociate from each other. The strand of messenger RNA is made during transcription include regions called exons that codes for a protein and non-coding regions called introns. In order for messenger RNA to be used in translation, the non-coding introns need to be removed and modifications such as 5' cap and 3' poly A tail are added. This process is called intron splicing and is performed by a complex made up of protein and RNA called a spliceosome. This complex removes the intron segment and joins the adjacent exons to produce a mature messenger RNA strand that can leave the nucleus through a nuclear pore and enter the cytoplasm to begin translation. And translation is a process which we will discuss in our next video. So if you want to find out that how this messenger RNA translates into a protein, you need to subscribe and share this